In this episode of Voice of the Sea, we're learning about Ciguatera poisoning, the seafood illness that comes from toxins produced by microscopic algae in warm, nearshore waters. Researchers in Hawaii are studying these tiny algae to better understand where they are most abundant and which species are producing toxins, with the goal of helping people eat safe seafood. We head out with Eileen Nally from Hawaii Sea Grant and her team of researchers to collect a sample and follow it through the process of counting and identifying the microscopic algae. But first, we hear from Program Manager at the Division of Aquatic Resources, David Sakoda, about his experiences with Ciguatera poisoning.
I'm a lifelong fisherman. I grew up pole and line fishing with my dad and brother, and then got into spearfishing and just learned more and more about the ocean. So that just gave me a passion for fisheries management and making sure that we have fish for the future. I had Ciguatera 3 times, a couple times back in 2006, 2007, and then once in 2021. The first time I had it, I had vomiting and just weakness, and then for about 3 months after that, my hands were just really sensitive to cold things, so I couldn't pick up a cold drink or touch ice or anything. The next time I got it, I had like weakness and body aches, so all the symptoms kind of aligned with what I knew about Ciguatera, and so I just self-diagnosed.
Do you know what kind of fish that you got the Ciguatera poisoning from?
Yeah, so the first one, I'm pretty sure it was from a table boss, a'awa. The second one was a big uku on Maui. The last time I got it, most recently, it was a roi that put me over the threshold. But I had been eating fish that I'd caught for about 5 days straight. So I ate a kahala, an uku, some uhu, a moana kali, and then the roi. And then after that, I just felt achy and my symptoms lasted for about a week.
And you're still fishing?
I still fish. I think those experiences have definitely influenced what I target. Try to avoid like the head and the belly portions. Yeah, just try to eat in moderation. I love eating fish. It's impossible to stop.
Has it changed where you fish?
Not really. I fish where there's fish. I fish where I know.
What about sharing the fish with your family?
I think about it more now. I don't share anything that I wouldn't eat myself. The time that I got Ciguatera, my wife also got it when we ate the roi. Fortunately, our 2 kids didn't get it. After that episode, it made me be more cautious about sharing fish with friends and family. The 3 times that I got Ciguatera, I never reported it, and I presume a lot of fishermen that do have Ciguatera don't report it. So I think there's a lot of information out there that we're just not collecting, and by collecting that information from fishers with that experience, we can just help build a better knowledge base and inform people about Ciguatera and help people make better choices.
Next, we're collecting a sample for analysis with researcher Justin Chang.
Today here we're at Waikiki and we'll be jumping in the water to grab some limu. When we collect the limu, we're looking for an epiphytic dinoflagellate, which causes Ciguatera poisoning.
What kind of limu are we going to be getting today?
We mainly look for 3 species. It's either Halimeda, Dictyota, or Lingbia. But if they're not in the area, we're really just sampling any predominant limu that we can find.
Are you targeting areas that people fish?
We are targeting areas people fish. Currently here, there's people fishing just down the rock walls. This is definitely a spot where people would want to know if Gambardiscus is there or not, so yeah.
Awesome, let's do it.
Yeah, let's get out there. So we went out there, we found, this isn't typically a limu that we collect, but if it's a predominant species, we'll collect it anyways. Gambardiscus is a epiphytic dinoflagellate, so it's anchored onto the limu. So to get it off the limu, we pour out some of the water in our Tupperwares and we give it a good shake to shake off all the Gambler discus, and then we'll pour our sample into our filters, and it'll pass through our filters, which have a 220 micron mesh. So hopefully the first mesh is going to get off any big debris like sand or rocks, and then we'll collect on the bottom mesh the Gambler discus.
So you're really not collecting the limu itself, you're really collecting what's living on top of the limu.
Correct. We're just collecting the dinoflagellates that are anchored onto the limu. After going through about 5 cycles of shaking it, we pop off the first layer, and we'll pour our filtered material from the mesh into our sample files, and we'll head back to the lab and look for Gambler discus under microscopes.
Wow. I can't wait to see what's there.
Yeah.
Now, Coastal Health Program Coordinator Kelsey Kunoshi prepares the sample for counting under the microscope.
So after we collect the limu and process it on shore, we bring it back to the lab. This sample comes from a Galaxura rugosa, otherwise known as pa kala kala. A lot of herbivorous fish and wrasses, surgeonfish like to eat this. This one is going to get used for plankton counting, where we're physically counting the Gambler discus, which is what produces the ciguatoxins that causes the Ciguatera poisoning. And some of it also gets subsampled for DNA, because not all Gambler discus species produce ciguatoxins. Other labs do the DNA work, which is faster and more accurate than what we'd be able to do under the microscope. And then a different lab does the toxin analysis. So I'm going to pipette 1 milliliter onto a gridded microscope slide, and then we'll count it in the lab.
So you're actually going to visually look?
Yeah, we're going to visually look. A lot of times we see a lot of cool diatoms, some biotic debris, larvae. Usually it's not just the Gambler discus, we see a lot of other stuff.
And that's all kind of living on the
limu, yep. Or it came from the seawater that we filtered.
About how big are the Gambler discus?
You can see them at 10x. So,
oh wow, they're big.
Yep, pretty big dinoflagellates. And then this is what our prep slide looks like. You can see how the debris has dispersed throughout. And then this is what we'll take to the microscope.
So you're just scrolling through the squares in an organized way?
Yeah, usually we go up and down. We start at a corner and we go throughout the entire sample. And then when we find one, we'll mark it on the tally. This can definitely take a long time, but it's important that we're really thorough with this.
Do you enjoy this part of the work?
I do. It's definitely really interesting to see what is in the area. Here's another one, this one's smaller. It's smaller, but you can kind of still see the color and the membrane. When I was first training, it would take me so long just to identify one.
Wow, that's neat.
So I did like maybe 2 or 3 rows and I just found 3. So this sample probably has a lot. Gambler discus and ciguatoxins have been around since before humans were in Hawaii. So this is something that's naturally occurring, and it's not something that was introduced. It's just endemic to the tropics, and some local traditional ecological knowledge, especially from Fiji and Vanuatu, have noted that after big rain events or extreme weather events, storm events, it's associated with Gambler discus or Ciguatera outbreak.
Oh wow.
Even the flowering of certain trees, fruiting of certain trees is associated with Ciguatera outbreaks. But we do have these trees in Hawaii, so I like to make note of that.
Which trees?
The wili wili haole, which is a erythrina, and we also have the kamani tree. Some studies have even suggested that when they read captain's logs and did carbon dating, the migration patterns correlated with Ciguatera outbreak events. It can be assumed that people were leaving these places out of necessity for food. So some of the things that I've been working on are like a pocket guide of limu in the area and fish that eat that limu, or like the anecdotal things about the flowering of certain trees, and even an educational module that we can use in school, beginning at the bioaccumulation, biomagnification issue, because that's what this is.
Bioaccumulation, biomagnification?
So bioaccumulation is the buildup of one substance in an organism over time, and then biomagnification is the buildup of the substance as it moves throughout the food chain. So if an herbivorous fish eats one ciguatoxin a day and it lives for 100 days, it'll have 100 ciguatoxins. But then let's say a bigger fish eats that fish, and then humans eat that fish, that's biomagnification. That's when humans will end up with the highest, potentially the highest concentration.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
You're watching Voice of the Sea. We're at UH Manoa in Eileen Nally's lab, learning about the Pacific Ciguatera Network and the research led by Talofa Fe'a in American Samoa. Let's talk a little bit about the issue. Like, what is Ciguatera? What is this poisoning?
So in the Pacific, Ciguatera is kind of our harmful algae bloom issue. It is the primary algae-related seafood-borne illness in the Pacific Islands. It is our regional issue, and it is profoundly impactful and incredibly understudied as compared to harmful algae blooms in a lot of other places that we might see in the continental US. You have these tiny little algae that are growing attached to bigger algae in the environment, and then fish go to eat the algae and it starts to accumulate in their system. So these toxins build up in fish over time or through the food web and higher in fish that eat other fish, for example. And then when people eat those fish, we can get quite sick. And Ciguatera poisoning can manifest in sort of a range of symptoms, ranging from sort of a mild flu lasting a day or two to really severe cases where people end up in the hospital for months, or in extreme cases, even death. It has sort of a suite of hallmark symptoms. One that people usually talk about is a temperature reversal, and cold feels hot, hot feels cold.
How are you working to categorize the Ciguatera poisoning, or you can try to make sense of this data from across the Pacific and these people's varied experiences?
Yeah, so we did surveys and interviews, and those are really important to sort of understand the impacts and the situation, because less than a quarter of cases in at least my past work are reported, and so most people aren't sharing their stories with a health agency or anything like that.
Do fish themselves get affected by the ciguatoxin?
Yeah, some of our collaborators at the University of South Alabama have looked at things like fish behavior in response to exposure to ciguatoxins, and they did see that when exposed to the toxins, the fish did behave a little bit differently, respond to stressors a little bit differently. It's an area that deserves a lot more attention and will be exciting to learn more about.
I want you to tell me about this workshop in the Republic of the Marshall Islands. How did their experiences compare with fishers here in Hawaii or in American Samoa, where you're also working?
They were very excited and interested to know what was happening with Ciguatera in the Marshall Islands, because it is an issue there, and not necessarily on the main atoll Majuro, but it kept coming up again and again that people living on other atolls that are more sparsely populated, more dependent on reef fish as a resource, had seen and had major issues with Ciguatera, and in some cases to the extent that people were afraid to eat fish. And so this was really impacting how they were able to eat, how they were able to fish and access resources, and so there's a lot of motivation there to understand what's happening.
Are you at a point where you can make recommendations to people? If I ask you, like, Eileen, where should I fish, or what kind of fish can I eat?
I would be hesitant at this point to make recommendations. I mean, we know that some locations and some species are going to be higher risk that I would recommend sort of limiting or being thoughtful before you eat them. For example, kole on the north shore of Kauai, many people have reported issues there, and so perhaps that would be something to steer clear of. Roi is something that's a pretty well documented cause of Ciguatera poisoning, and there's other species that certainly people do tend to get Ciguatera from, but is it unanimously in every location, every fish of this species you're going to get it? Not necessarily. And so it's always a balance, you know, when you tell people not to eat fish, they're going to eat something else. But we do know that certain parts of the fish are going to be higher risk. So the fatty tissues, the belly meat, the brains, the eyes, we know if you eat that, you're going to be more likely to get Ciguatera or have a severe case. That's certainly one easy way people can sort of mitigate the risk.
Right now, I am working full-time with Hawaii Sea Grants in Dr. Eileen Nelly's lab as the Ciguatera project's lead and coordinator on the ground in American Samoa. Ciguatera is known to be widespread in the Pacific. I think it's one of those places that have always been endemic to the disease, and now it's starting to be more widespread to other places who have never had it. And that's because these dinoflagellates, they really like calm, warm, tropical salinity waters, and that's the Pacific Ocean pretty much. Historically, it's always been the problem, but as our waters start to get much more warmer, it could increase cases of Ciguatera. Part of my work is to lead the research side of things, where we're conducting interviews, surveys, and also some sampling on the ground. We've also hired and have onboarded 3 amazing interns, 2 of them from the American Samoa Community College, and the 3rd one is a returning resident of American Samoa. The first and last documented study of Ciguatera poisoning in American Samoa, it was conducted by the local Department of Marine and Wildlife in 1997. So that's about 28 years ago. We're technically going back and revisiting that conversation with the people. Our red flag species, I like to call it, we have the mu, which is in American Samoa, it is the red snapper, the Lutjanus bohar. A lot of fishers have shared that this species have caused Ciguatera for them, whereas others have shared that they have managed to avoid Ciguatera poisoning their entire lives by avoiding to eat the species.
What methods are they using to check their fish?
The most credible that I've heard so far is our fishers sometimes try out small piece of the fish, see if they feel some symptoms, and if they don't, they proceed to eat the whole thing. Yeah. In partnership with the American Samoa Community College Land Grant Research Division, we're able to actually collect samples in American Samoa and take them in a lab just like this and analyze those samples and count them.
Tell me what kind of samples you're collecting.
Yes, so we try to target the most dominant algae species within a certain area, which would look different for each bay and each beach we decide to sample. We often look at Dictyota, Helmeta, and all of those limu.
What else have you learned from the surveys and interviews?
Ciguatera poisoning is very place-based in a sense, where I talk to one fisher from a certain village where they talk about the mu and they think it's ciguatoxic, and I talk to another one from another village and they're like, no, our mu is safe. So it's very place-based in a sense that Ciguatera might look different, not only among islands, but also a more local scale, like villages and bays.
We are looking for a few heroes, mentors, trailblazers, innovators, a passion to change lives, spark curiosity, open hearts, and awaken minds, help students answer the question, who am I? This could be your calling, but this is no job. It's the journey of a lifetime. Be a hero. Be a teacher.
You're watching Voice of the Sea. We're at Hawaii Pacific University's Waimanalo Makapu'u campus with Matthew Iyaki, whose lab is using new technologies to identify the species of tiny algae that produce Ciguatera toxins in our nearshore waters, in hopes of understanding and predicting when and where fishes are safe to eat. Okay, Matt, talk me through the process. They've collected the limu.
Yeah, so they collect the limu and then they shook all the water off of the limu, right? And so then we take that water and that's what we use for everything else. So you guys took some, you saw the students looking under the scopes, right? So that's one subset, and that's to get the morphological IDs and the counts under the microscopes. And then for what we're doing in the molecular lab is trying to get down to species level. And so to do that, this would be like a field collection from a couple of sites, and then they just get stored in these 2 mil tubes. They're in like a DNA preservation buffer so they can actually stay at room temperature. For the next step, we want to extract the DNA.
And is that going to give you the DNA just from the
Gambierdiscus?
Gambierdiscus?
No, it's going to give us like the DNA of everything that's on there. So this is what they call eDNA, or environmental DNA sample. So when we're pushing stuff through this filter, we're collecting whatever DNA is in the environment from different organisms, cells, from like even just DNA that's floating around in there. And then when we're selecting out like certain organisms that we want to pick up in that DNA, that we're doing what's called meta-barcoding. So we're getting this one selected fragment that's called the barcode, just like if you barcode in the grocery store, right? So it's like you run the barcode and you know like this is avocado, this is breadfruit, you know, this is, so it's the same idea. We're using this fragment and they call it the barcode to identify the organisms specifically to genus or species.
How many species of the Gambierdiscus are you generally finding in your samples?
We honestly don't know. So we don't know how many species are here, and that's kind of part of the project. Work that's been done in the past has identified at least like 3 or 4 species in Hawaii, and then we kind of know generally like from work throughout the Pacific that like which species are generally in the Pacific. But that's one of the things we're really interested in, is because under the scope you can really only identify the genus. The molecular techniques is really what's going to get us to the next level of saying like these are the animals that are here, these are the species that are here. And it's super important because like not all of them are as toxic. And so that's the other thing that folks have figured out in the last like 5 years or so, is that there's variation in toxin levels across these species.
And then once you've identified which species are present in our waters, what's your next step?
So I think when we identify, we want to look at the distribution, right? So that's why part of the sampling around Oahu, we now have samples around much of Oahu. And then I think focusing on Kauai next and the outer islands if we can get funding. And then that way we can say like, okay, where are each of these species distributed? And then we can just run these analyses and say like, hey, you know, you have really high levels of this species in your area right now. So, and then we can kind of set up like a warning system. I think eventually, you know, we're going to get to one of those quick tests that's fast, just because, I mean, like the science moves so quickly. Like even like these techniques weren't available 5 years ago. And so I think because of that rapid movement, as long as we keep investing in science and like, you know, investing in these kind of projects, like science that impacts communities, I think we'll get there. I just don't know when. The fishers have the right goal, right? It's like, we want to be able to know if this fish is good to eat or not as soon as we get it out of the water, and not wait at all, and you know, and be able to take that food to our family and know that they're going to be safe and we're going to be safe.
Healthy fish and safe seafood are critical for healthy coastal communities. Share your Ciguatera experience and contribute to the research. Learn more about Ciguatera research in Hawaii and the Pacific Ciguatera Network on our website at voiceofthesea.org. Follow us on social media at Voice of the Sea TV. Mahalo for watching Voice of the Sea. Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org.